microscope is so sensitive, they even can quantify the single molecule inside a cell. So in a way, we can just look one component inside individual cell. But as I mentioned, actually that's not fair, because if you just look one component, maybe you quantify it very well, but on the other hand, maybe another 200 components actually are different inside the cell, and we still don't know. So that part is the limit of biology. So we have no way to monitor all the components at the same time. And I think that's the limit, because, I mean, currently there's a technique try to develop like mass spec. They want to quantify like protein molecule, different protein molecule of one single cell, all the protein at the same time. That's what people try to develop. So if that works, maybe you can monitor multiple proteins at the same time. But at the current stage, we only can look very limited number of the protein, which is just maybe one or two at one time. And I don't think that's good enough for physicists, right? Because you will say, okay, yes, this one is noisy, but how about the others? We don't know.
Well, the kind of problem physicists deal with are usually multiple. In your parlance, the typical typical system physicists are dealing with actually. I was about to ask you whether you always just focus on a single cell or like a multiple cells simultaneously, because usually that's the type of problem physicists are dealing with. And in fact, like when you say quantifying what kind of state this a single cell is actually in, that usually is not. I mean, you don't have a sufficient number of samples to actually say what's the. Well, usually I guess what Siwei might be leading towards is that, well, okay, can we formulate this in some kind of probabilistic problem that, if there are, three possible states with equal odds, so it's like 1/3 each, but then you can't really say that if you you're only observing a single cell, right? So
well, but the number I think may not be that problematic, because
is that right?
now we can monitor like 1,000 cells at one time. That's doable.
Okay.
But the problem is, again, for every individual cell, we can only monitor one or two components, and we have to control.
But the rest of other components, it may have very little effect. It could be a small perturbation.
That's what we will assume.
What you are doing.
But nobody knows, right?
Right, but so that will be the next level of a quantified component.
Yes, yeah.
I mean,
and we try our best to control the conditions, so hopefully the other component will be more constant. That's what we can hope.
Because basically the mathematical tool for the stuff you are dealing with is something called stochastic calculus, right? And time sequence. So, but usually you need to have some control of the variable, and this is akin to doing the perturbation in theoretical physics. So. So there are some people starting to do that, right? I mean,
yes.
In Princeton, there are a bunch of people.
Yeah, I mean, people start to get into this issue, and actually a lot of physicists actually are involved. Yeah.
Can we get our panel members come forward? Then we'll start the panel discussion. So you can ask your question later on.